In the lab, we're mainly focused on melanoma and the genetic underpinnings of this disease. Melanoma is a very terrible disease. It is not the most common skin cancer, but by far it's the one that causes the most skin cancer deaths. And over the years, melanoma therapies have been pretty poor. But in the last 5 to 10 years, many new melanoma therapies have come online, and these have had a major impact on patients. However, many patients never respond to these therapies, and many that do, they develop resistance to these therapies. So there's still a great need in the melanoma field to develop new therapies, and we are interested in identifying new genes that cause this disease, where each of these genes may represent a possible therapeutic opportunity.
Sequencing analysis of several patient melanomas has identified several kinds of genetic changes in the human melanoma genome. One of these changes are copy number variations. In regions of copy number variations, you have genes that have more number of copies or less number of copies as compared to a normal genome. Analysis of the human melanoma genome has identified that a lot of genes undergo such copy number variations. Now, only some of these genes are important for disease progression, and we call them drivers. The rest of them are randomly altered passenger genes that do not have any effect on disease progression. It is really important to find these disease-promoting driver genes that are being altered, as it does two things. First, it helps us understand the biology of the disease better, and two, it helps us identify novel targets for therapeutic intervention of melanomas. But the problem is, amongst a huge range of genetic changes, how do you distinguish driver genes from passenger genes? In order to do so, we used a zebrafish model of melanoma. This model uses a recurrently altered BRAF oncogene along with a loss of function p53 tumor suppressor gene. Combination of these mutations causes all the zebrafish to get melanomas. The idea is, if you look at recurrently altered genes in the human melanoma and compare it to recurrently altered genes in fish melanoma, the genes that are commonly amplified would be driver genes that are essential for disease progression. In doing so, we identified about 374 genes that are commonly amplified in human and fish melanomas. And on further integrative genomic analysis, we identified GDF6 as a very important recurrently amplified melanoma oncogene.
We identified GDF6 as a BMP ligand, so it's a bone morphogenetic protein. And we've done a number of functional tests to assess whether it has some role in melanoma tumorigenesis. So in one series of tests, we have knocked it down, and what we found is that when we knock down GDF6, tumors arise later and they grow more slowly. And in another series of experiments, what we've done is to overexpress GDF6, and in those circumstances, what we find is that the tumors arise more quickly and they grow much more quickly as well. We've done mechanistic characterization of GDF6, and in a number of different genomic analyses and functional studies, we found that GDF6 acts in a couple of different ways to affect melanoma cells. So one of the things that it does, it appears that GDF6 during the course of melanoma genesis is amplified at the chromosomal level. And what this does is to increase the amount of GDF6 that is present in melanoma cells and can act on melanoma cells themselves or neighboring melanoma cells. And when it does that, what it can do is it can affect the expression of genes downstream through bone morphogenetic protein signaling pathways. And one of the genes that it affects is MITF, so it can turn off MITF, and this gene is a master regulator of melanocyte differentiation. So what GDF6 does is it prevents the differentiation of melanoma cells and keeps them in a state where they can divide more and more. In addition, what it does is it represses the expression of another gene called SOX9, and what that has the effect of doing is it keeps these melanoma cells alive. So in acting through this BMP signaling pathway, GDF6 not only prevents differentiation, but it also prevents the death of melanoma cells.
To see if GDF6 is clinically important, we next analyzed expression of GDF6 in several patient melanomas. What we identified was that about 80% of human melanomas express GDF6. We then compared patients whose melanomas had high GDF6 with patients whose melanomas had low GDF6. In patients where melanomas had high GDF6, there was an increased risk of metastasis and there was a shortened survival of those patients. Conversely, patients whose melanomas had less GDF6, there was a lower risk of metastasis and a higher chance of patient survival. In sum, there are a couple of very important points. One is GDF6 is expressed in a major fraction of human melanomas. And two, melanoma cells are dependent on GDF6 for its survival. So you get rid of GDF6, melanoma cells do not survive. This makes GDF6 and components of the BMP signaling pathway really important for therapeutic intervention.